Hi, I'm going to walk you through the cell division Gizmo lab. So let's take a look. Okay, these prior knowledge questions are asking before cell division happens, what do you need to do? What does a cell need to do so that you just don't get smaller and smaller cells? So hopefully you know that. Also, same thing, what does the DNA need to do before it splits into two so that each new cell is not just getting um, half of the DNA? So if you're not quite sure, you'll actually find this answer later in the lab, so you could come back. Okay, so let's take a look at the warm-up. It says to set the cycle length to 12 hours, which means every 12 hours the cell will divide. And then it says to click play until the maximum number of cells is shown and then pause it. So let's go ahead and do that. And actually I should be over here. This is what it will look like when you open it up. So I'm gonna go ahead and push play. And these are the hours that are ticking by, obviously speed it up. And then here is every 12 hours, a cell is dividing. And of course, the more cells you have, the more cells that are going to divide. So it's definitely getting more cells. Okay, now we've hit the maximum number of cells. So I'm going to go ahead and pause it. And it says click on the magnifying. So actually, I already had that clicked. I'm going to click on that. And then now if I'm pointing to a cell, it will magnify it a little bigger so that I can see a little bit more of what's going on inside the cells. Okay, so this one I can see the little chromosomes. Here I can see them all lined up. Here I can see them kind of all condensed. Here there's actual chromosomes that have coiled up. Um, so of the 100 cells shown, how many are in the process of dividing? So I think you sort of can eyeball it, or you can go to the next question and go back and answer that because it's going to tell you, but it says click on the bar chart. So if you click on the bar chart, oops, then you are going to click on show numerical values, which I already had, and then it can tell you how many cells are in the different phases. So here's interphase, and then here are all the other phases. So interphase is when it's not dividing, and then these are the phases of mitosis where the cell, in particular, the nucleus is dividing. So you can go back and sort of answer that question by just adding these up, and you can, that would be how many cells are in the process of dividing and the rest are not in the process of dividing. Okay, so then you can answer those questions. And then we're ready for activity A. So activity A, we're going to go ahead and reset everything. And we're going to go back to the description tab. And then it says click on the right arrow once so the interface is shown. So it was originally this picture, and this shows all the phases. And then it's going to highlight one phase at a time. Okay, and then it says click play and hold the cursor over the cell. Observe the cell as it divides several times. And what do you notice happening during this process? So let's go ahead and click play, and then we'll hold our cursor over here so we can see. So we can, so we can see these stages happening super fast. So you notice like it's interphase for a long time, and then all the other ones happen really fast. Okay, and then there it goes, just all the cells continuing to divide until they fill up the page. So we're going to go ahead and summarize each cell. So you're going to have to sketch it. You might have to print out this paper and do a little sketch of it. You might be able to copy and paste a picture of this. But the summary I'd like you to write in your own words. So for example, don't copy and paste this description. Um, Read it over and just pull out the main points. So right here, the cell's growing and maturing. So there's the growth phase, which is G, and then the chromatin, which is all of this genetic material, is duplicated because remember, before it splits, the DNA has to be duplicated so that each new cell gets the exact same number of the original DNA. So if you had 46, then it needs to copy those 46, and that way each new cell gets 46. If you don't copy the DNA, then each new cell is going to only get 23, and then when that one splits, it's going to be 12 and a half, which is going to end up being some messed up cells. So the chromatin has to copy itself, duplicate, and then the other organelles need to make a copy of themselves too, so that each new cell gets the same number of organelles as the original cell. So that's what you can put for your summary. You can say that the cell grows and matures, and that the chromatin makes a copy of itself or duplicates itself, and then the organelles make a copy of themselves. Then you're going to go on to each of the phases and just summarize the main points. Okay, so what's happening in prophase, and you can see that what the chromatin, the DNA used to be all uncoiled, like tangled up thread or stuff like that, and then they coil up into distinct shapes, which we call chromosomes. So chromatin and chromosomes are exactly the same. They are DNA of the cell, but chromosomes are just coiled up DNA. Okay, so you're going to go ahead and finish those stages and answer the questions. What are the 4 phases of mitosis? So obviously, mitosis is going to be, this is the first phase of mitosis. Okay, so interphase is actually not considered mitosis, but starting with prophase, okay, those are the 4 phases. And then this part, so mitosis is just the making a copy of the nucleus. So once that's over, this other phase, this is just kind of where the rest of the cell splits. The nucleus has already been copied and separated. So mitosis is over, and this doesn't count as mitosis. This is where the cell pinches in two. Okay. So now you can answer that question. And if you weren't able to answer the first 2 questions at the beginning of the lab, you can go back and answer that as well. Okay. Now let's go ahead and look at activity B. So we're going to reset, and we're going to select the table tab. And this time we're going to set the cycle length to 10 hours. So every 10 hours, the cell will divide. And then we're going to play it, pause it, and then we're going to record the data once we hit the maximum. So let's go ahead and let that play out. So every 10 hours, now it's at 10, it divided. At 20, so those 2 divided. And the more cells you have, the more cells they'll make. Okay, so let's go ahead and see that. And then now we have the maximum, so we'll go ahead and pause it. Okay, and then we're going to go ahead and click record data. So record data. And then we're going to go ahead and record the number of cells that are in each of the phases. So 83, 10, 3, 1, 2, 1, we're going to go ahead and put that in here. 83, 10, 3, 1, 2, 1. And then we're going to, and then we'll reset it. So reset it and then play it again. Okay, and we're going to do 4 trials, because remember, you always need to do more than one trial, as many as possible to get an average. So we'll do that. Let that run until it hits the maximum. And then pause it. We'll record our data. And we'll go ahead and write how many cells were in interphase, how many were in prophase, how many were in metaphase, anaphase, telophase, cytokinesis. And then we'll go ahead and take the average by adding up the number in interphase, dividing by 4. And so this is like a snapshot of where all the cells are at the point where we kind of pause this simulation. And you can see most of them are in interphase. Okay. And so it seems like the cells are spending the most time in interphase. And then if we only see like 1 or 2, like 1 in cytokinesis, that must happen really fast, because when we say freeze and we capture that picture, we're hardly seeing any in cytokinesis. We don't see any in anaphase, so that must be a really short phase as well. Anaphase is when the chromosomes are moving apart, so that happens really fast. Okay, so we're going to go ahead and use your data to calculate how long each cell cycle is. All right, so we're going to go ahead and estimate. Once you find the percentages, then you're just going to multiply that by 10 hours. And so when you multiply by 10, you're basically just 8% would be 0.08, and then you would just move that decimal over 1, so it would be 0.8 hours. If you have trouble, you can email me and I'll help you with that. Okay, so the last part is to look at a graph. Okay, so we're going to go over to the graph tab, and we're going to set the cycle length to 5 hours. So they're going to divide even fast in even shorter of a length of time. So every 5 hours, they're going to divide. And then we'll go ahead and play. Okay, so go ahead and play. Okay, and then stop. Now your graph, I did this already. It says to click on the negative button. Your graph should look like this, okay? And then it says to click the negative button until the whole graph is visible. So you want to zoom out until you see the whole graph. Right here, it tells you that now you see the whole graph. Okay, so somehow you're going to sketch this. Again, you might have to print this or figure out a way to draw it on your PDF. And then explain what the general shape of this is. And if you've done a little bit of algebra, you might know that this is like an exponential curve, called an exponential curve. But you could just say that it seems to curve upward really steeply. Okay, and then you're going to answer these questions based on your graph. How long did it take to grow the first 20 cells? So here, to get to 20 cells, it seemed to take about, so here's 20 hours, here's 30 hours, here's 25 hours. So like a little less than 25 hours, maybe like 23 hours, 24 hours. Okay, and then the last 20 cells, So from here to here, how long did this take to get from 80 to 100 cells? Well, it's very, very short. So this is like 5 hours. It was definitely shorter than 5 hours, maybe like an hour and a half, 2 hours, hour and a half. Okay. Okay. And then the last question is, what do you think is going to happen if cells just grow out of control? If this cell cycle, it starts dividing faster than it should, um, and you start making too many cells, what do you think could develop, or what do you think could happen if your cell cycle is not regulated? Okay, so hopefully that helps. If you have any questions, please email me